Prior to HeLa cells, there had been various failed attempts to grow human cells outside the body, resulting in a limited understanding of human cells and how they operated. HeLa cells were extracted in 1951 from a cancerous tumor, and shortly after it became evident that these cells are unique in many ways. It had abilities to endlessly grow and multiply. It was an incredible discovery, and it didn't take long for these cells to be shared to researchers around the world. Scientists were able to use these cells in diverse ways, opening a whole new door of possibilities. By cultivating and studying these cells, scientists could develop various techniques and treatments for many different viruses and mutations. Overall, these cells played a critical role in medicine and vaccine development. It was in 1936 when a polio epidemic occurred throughout regions in the United States. Polio was an extremely serious disease, targeting the nervous system and resulting in paralysis and occasionally death. Although surviving the disease was a possibility, long-term symptoms included deformed limbs, artificial respirators, and paralysis in extreme cases. It became increasingly important that a vaccine was developed for the spreading disease, and so the hunt for a cure began. Due to the fact that HeLa cells are able to easily get infected by polio and can be produced in large quantities, scientists were able to use these cells to test for a vaccine. This is just one example out of the hundreds of other ways that HeLa cells have been used.
In general, HeLa cells can be applied to multiple areas of study. First by obtaining the cell and cultivating it in vitro with a nutritive medium. Then, depending on what is planned to be done with the procedure may change. Specifically for poliovirus, after the cells are cultivated, they can be used for cultivation of the poliovirus by infecting them with any of the 3 types of poliomyelitis virus. The diagram depicts how HeLa cells were used to develop a more recent virus, COVID-19. As you can see, researchers are able to adapt the HeLa cells, generating them to evolve specific receptors in order for the virus to access the cell and begin the infection cycle. This cycle of infection allows researchers to understand how the virus affects human cells and alters or impairs its functions, as well as monitoring how fast it spreads. In the specific case of COVID-19 virus, HeLa cells provided an understanding on molecular processes of the virus itself, as well as the various pieces required for infection.
Although HeLa cells have led to many discoveries and advancements in science and medicine, the backstory behind these cells is quite controversial. These cells were taken from Henrietta, a woman suffering from cervical cancer. These cancer cells were taken from her without consent, and unfortunately, no confidentiality was associated with these cells, as her identity was made known in all medical files. In addition, the Lex family struggled financially for years, and yet no financial benefit was given to his family. In fact, the family was not notified about these cells until the mid-1970s, around 20 years after the cells were initially harvested. Despite this upsetting backstory, the specific application of HeLa cell technology includes various vaccines, treatment for cancer, and contagious viruses. HeLa cells were also the first cells to go to space and were used to determine how cells interact in zero-gravity environments. In other words, Henrietta and her family were treated unjustly when it came to harvesting and the confidentiality of the use of her cells. And yet, it is hard to ignore the many advances in science that were made.
Fortunately, there have been many adjustments and additions to the previous rules when it comes to bioethical standards since the 1950s. In the past, consent was never considered. Therefore, many patients became donors while being completely unaware that cell and tissue samples were being taken from them. Regulations on how cells were used was completely overlooked. Research projects of whatever kind could take advantage of these cells and do whatever they pleased. Furthermore, patients had no rights when it came to reviewing their medical records or even being able to hold their own copy. Presently, all of these past issues have been eliminated with the implications of new laws. In order to harvest cells, patients now must undergo an incredibly strict and thorough consent process in order for any donation to occur. This process allows the patients to be completely informed and comfortable with the decision they are making before continuing, an option that Henrietta Lacks never had. In addition, examining research studies prior to starting them is now a standard practice. This ensures that the planned research studies are valid and will appropriately use the cell sample. Medical records are given to patients, as patients now have the right to view them as well as have their own copy. Although the lack of rules in the past has led to many unethical and controversial situations, it has only improved since then. Currently, cell research is much more just, and patients now have the rights they deserve.